Okay, so here's something wild. About 25% of the global population is estimated to have metabolic dysfunction associated steatotic liver disease, or MASLD. That's 1 in 4 people, and most of them don't even know it.
That's a staggering number. I mean, we're talking about a condition that's not just common, but also sneaky. Most people don't even realize they have it until it's progressed. So, what's driving this rise in MASLD?
Well, a lot of it ties back to lifestyle factors like diet and exercise, but what's really fascinating is the role our genetics play. There are specific genetic variations that can make some people more susceptible to MASLD, and understanding these could completely change how we treat the disease.
That's such an exciting idea. Precision medicine for something as widespread as liver disease. So, what are the key genetic players we're looking at?
There are 5 big ones: PNPLA3, TM6SF2, MBOAT7, GCKR, and HSD17B13. Each of these genes has variants that either increase the risk of MASLD or, in some cases, actually protect against it. Let's start with PNPLA3 because it's probably the most well studied.
All right, let's hear it. What does PNPLA3 do, and how does it contribute to MASLD?
PNPLA3 is a gene that provides instructions for making a protein involved in breaking down triglycerides, which are a type of fat. The problem comes with a specific variant called RS738409, or the I148M variant. This variant doesn't just stop the protein from working, it actually changes its behavior. Instead of breaking down fats efficiently, the protein starts to accumulate in lipid droplets inside liver cells, leading to fat buildup.
So, it's not just a loss of function, it's like the protein is actively doing the wrong thing.
Exactly. It's what researchers call a gain of dysfunction. And here's where it gets even more interesting. People with this variant might actually have reduced fat production in the liver, but they also experience increased fat breakdown through beta oxidation. It's a paradox, but the end result is still an unhealthy accumulation of fat in the liver.
That's fascinating. Now, what about HSD17B13? I've heard this gene is a bit of an outlier because its loss of function variants are actually protective. Is that true?
That's absolutely correct. HSD17B13 is involved in processes like steroid hormone signaling and managing lipid droplets. But here's the twist. Certain loss of function variants, like RS7261357, are associated with a lower risk of MASLD, less fibrosis, and even a reduced risk of hepatocellular carcinoma, which is a type of liver cancer. Researchers think this protective effect might be linked to changes in retinol metabolism and reduced inflammation.
So, if losing the function of this gene is protective, does that mean targeting HSD17B13 could be a potential treatment strategy?
Precisely. In fact, several therapies aimed at inhibiting HSD17B13 are already in clinical trials. The idea is to mimic the protective effect of these loss of function variants by pharmacologically reducing the activity of this gene.
That's exciting. What about TM6SF2? How does it fit into the picture?
TM6SF2 encodes a protein involved in the secretion of very low density lipoproteins, or VLDL, which are particles that transport fats out of the liver. A specific variant, RS585422926, reduces the levels of this protein and impairs VLDL secretion. As a result, fats accumulate in the liver, increasing the risk of MASLD and its more severe forms, like fibrosis.
So, if the liver can't export fat effectively, it just ends up storing it?
Exactly. And this variant doesn't just affect fat storage, it's also associated with higher levels of ALT, a liver enzyme that indicates liver injury. But here's the kicker. This same variant might actually lower the risk of cardiovascular disease because it reduces the amount of atherogenic lipoproteins circulating in the blood. It's a classic double-edged sword.
That's a tricky balance. Let's move on to MBOAT7. What's its role in MASLD?
MBOAT7 encodes an enzyme that's involved in remodeling certain types of lipids called phosphatidylinocytols. A specific variant RS641738 reduces the expression of this enzyme in the liver. This leads to an increase in free arachidonic acid, which is a precursor to inflammatory molecules. The result? Increased liver inflammation, fibrosis, and a higher risk of hepatocellular carcinoma. However, the effect size of this variant is relatively small compared to others like PNPLA3.
So MBOAT7's impact is more about inflammation than fat accumulation?
That's right, it's a smaller piece of the puzzle, but still an important one.
And what about GCKR? That's the last one on your list, right?
Yes, GCKR is another fascinating gene. It encodes a protein that regulates glucokinase, an enzyme critical for glucose metabolism in the liver. A variant called P446L reduces the function of this protein, leading to increased glucokinase activity. This overstimulates glycolysis, the process of breaking down glucose, and produces more malonyl CoA. This molecule is a key driver of de novo lipogenesis, or fat production, in the liver. The result is higher triglyceride content and ultimately steatosis.
So it all ties back to these interconnected pathways: lipid metabolism, inflammation, glucose regulation. It's like a web of interactions.
Exactly. And it's not just these 5 genes. For example, MTARC1 is another gene that's been found to have a protective variant. This variant improves mitochondrial function and reduces oxidative stress, which can help prevent MASLD.
This is such a complex but fascinating area of research. How does all this genetic information translate into potential treatments?
That's where things get really exciting. By understanding a patient's genetic predispositions, we can start to predict how they might respond to different treatments. For instance, studies suggest that the PNPLA3I148M variant might influence how well someone responds to lifestyle changes or certain supplements like omega-3 fatty acids.
So we're talking about personalized lifestyle recommendations based on someone's genotype?
Potentially, yes. And when it comes to medications, genetic variants can also affect how effective certain drugs are. For example, GLP-1 receptor agonists and FGF21 analogs, which are being explored as treatments for MASLD, might work differently depending on a person's genetic makeup.
Are there any therapies being developed that directly target these genetic pathways?
Absolutely. One promising approach is RNA interference, or RNAi, which can be used to silence specific genes. For example, therapies targeting the mutant PNPLA3 protein have shown promising results in preclinical studies. Similarly, RNAi therapies are being developed to inhibit HSD17B13, mimicking the protective effect of its loss of function variants.
That's incredible. What's next for research in this area?
The next steps involve using integrative multi-omics approaches to better understand how these genetic variants influence metabolic and inflammatory pathways. New technologies like single-cell and spatial transcriptomics could help us see how these variants affect different cell types within the liver. And of course, pharmacogenomic studies are needed to determine how genetic risk profiles can guide treatment decisions.
It sounds like we're on the brink of a new era in liver disease management, one where treatments are tailored to each individual's genetic makeup.
That's the goal. By incorporating genetic screening into routine care, we can identify which patients are most likely to benefit from specific interventions. It's an exciting time for this field, and there's a lot of potential to improve outcomes for people with MASLD.
This has been such an enlightening discussion. Thanks for breaking down such a complex topic into something so accessible.
My pleasure. It's always great to dive into these topics and explore how science is reshaping medicine. Thanks for tuning in, everyone. We hope you found this as fascinating as we did. Until next time.